Welcome everyone, I'm Eva Viesi, and today I will present you some computational methods to preprocess single-cell RNA sequencing data. The first part of the webinar will be dedicated to data loading, filtering, normalization, and identification of highly variable features, while the second part, presented by my colleague Simone Avesani, will be related to data scaling, dimensionality reduction, batch effect correction, and integration. So, the data that we will analyze comes from the GEO platform, which is a large publicly available resource for gene expression and sequencing data. And GEO contains many types of experiments, including microarray-based studies, RNA-seq, ChIP-seq, and ATA-seq experiments. Researchers can upload their dataset to GEO along with metadata describing the samples, experimental conditions, and preprocessing methods. Each experiment in GEO is assigned a unique accession number, which can be used to search, retrieve, and download the data. In GEO, there are various file formats which are used to efficiently store single-cell RNA sequencing count matrices. And these matrices are typically very large, with genes features as rows and cell barcodes as columns. And one of the key characteristics of single-cell RNA sequencing data is that most of the values are zeros, as we can see in this matrix, and this is a phenomenon known as data sparsity. And this sparsity may occur for several reasons, such as low sequencing depth or some technical limitations in capturing RNA molecules from all the cells. And popular tools to analyze this data are SERA and SCAMPy. And these tools provide useful functions to load and analyze raw count matrices. And because of the majority of values in these matrices are zeros, SERA leverages sparse matrix representations whenever possible, resulting in significant memory and speed savings. So the preprocessing pipeline of single-cell RNA sequencing data involves different steps. And these steps include the quality control of the data and filtering, the normalization, the detection of the highly variable genes, the scaling of the data, and finally the batch effect correction. The goal is to have data clean and standardized for then subsequent analysis and tasks. So the first step of the pipeline is the quality control and filtering of both low quality cells and low expressed genes. Specifically, we want to evaluate the total number of unique molecular identifiers per cells, which represent the original mRNA molecules or the original transcripts. In this case, we want to identify both anti-droplets that show extremely low counts and putative doublets, which instead can have extremely high counts. Additionally, we evaluate the number of detected features or genes per cell to filter out cells that have few expressed genes, as well as genes that are detected only in a small subset of cells. And finally, we can also assess the percentage of mitochondrial and ribosomal RNA, since a high abundance of these transcripts may indicate stressed or dying cells. So first, we can, for example, calculate and visualize specific statistics for each cell, such as the number of unique molecular identifiers or number of counts per cell, and the number of detected genes, that are those genes that have at least one read count per cell. Here in the violin plots, each dot represents a cell from a specific sample. And here, after applying a mild filtering, we retain, for example, only the genes expressed in at least 10 cells. The scatter plot to the right shows the correlation between these two variables. And what we expect is to have and high correlation between N counts and N features, because this indicates that cells with a larger total number of reads are associated with more genes that are detected, and this typically corresponds to high quality cells. So, as mentioned earlier, low count and low features detected are likely to indicate antidroplets or broken dead cells with very low RNA content, while high count and high number of features may suggest possible doublets, so cells that have been merged together, or multiplets, and they show too much RNA content. So these are the same plots after applying filtering. A suggestion here is to avoid filtering too aggressively, as this may remove very rare cell populations. And for instance, in this case, we filtered out cells having fewer than 500 total UMIs, 200 detected genes, and more than 15,000 total UMIs and 4,000 detected genes. So to remove anti-droplets and to identify potential doublets. And as we can observe, there is still a strong correlation between the two variables, and this correlation has slightly increased. Finally, we can also compute and visualize the percentage of mitochondrial genes by searching for the NT pattern, for example, and the percentage of ribosomal genes by searching for the RPSL pattern, which corresponds to mRNA coding for ribosomal proteins, so the small and the large subunits. And as we mentioned previously, a high percentage of mitochondrial genes, so higher than 10-15%, may suggest stressed or dying cells, while ribosomal genes aren't always filtered, but unusually high levels could indicate artifacts or poor quality cells. In this case, based on the observed percentages in this sample, we filtered out cells with more than 20% of mitochondrial and ribosomal reads. However, in this case, the ribosomal content, as we can see in the violin plot, was not high enough to indicate low quality cells or some technical artifacts. So now we will walk through these first preprocessing steps in R using the Sera package. So let's switch to R. Okay. So first we need to load the required libraries. So these are the libraries that we are going to use. And then, so after the count matrices have been generated, the next step is to analyze this single cell RNA sequencing data. And the R package Sera is one of the most used toolkits to this aim. So before creating the Sera object, we can use several functions to load and manage different single-cell RNA sequencing file formats. For example, the HDF5 file can be read with this function using these parameters, in particular, so the file name. The MTX file format can be read with this function by passing the name of the count matrix together with the cell barcodes and the name of the features or the name of the genes. Or alternately, we can use the read10x function by passing the directory name containing all these files. We can also load a .lum file using the connect function and casting it to a Sera object. We can use the read RDS function to load an RDS file, which is typical file format for R. And here we are using the name of the file. And finally, we could have this annotation data object that usually is used in the ScanPy Python package. And here we can convert the file name to an H5 Sera file, and then use the function load H5 Sera to read the data. So finally, we have all our count matrices, and we can create the Sera object. So now we are going to see an example. So we downloaded data from the Geo platform. In particular, so this is the PMID, and this is a single cell RNA sequencing experiment of peripheral blood mononuclear cells of 7 samples with COVID-19 and 6 age-matched healthy individuals. For our analysis, so to speed up all the analysis, we selected only 3 patients and 3 healthy controls. So we first start by specifying the path to the count matrices, that in this case are 6. So we can specify the path and we list the files present in the directory. And then we can load data into R and generate our sparse matrices. In this case, the files are all in RDS format, so we can use the read RDS function. So we are applying to our list of files this function. Okay, And then to recognize which is the sample from which these matrices come from, we will assign a name to each count matrix. And so we will use this function to assign our sample names. So here we can see that we have the name of the sample, and we can also observe that this mat.files object contains 3 count matrices that have been generated by the DropEst toolkit. And these 3 count matrices are exon, intron, and exon-intron spanning reads. And usually and typically, since the exonic grids are the ones used in single-cell RNA sequencing to quantify gene expression, we select only the exonic counts for each sample. So we can have a look to the first 20 genes and 20 cells for one sample. Okay, and so we can see that this matrix contains many dots that represent 0 values in this sparse matrix representation, and only few integers that indicate the gene counts per each cell. Cell names are in the columns, while gene names are in the rows. So now that we have our count matrices, we can initialize a Sera object for each sample. And we start by applying a first mild filtering using this parameter, min cells, in which we want genes, so we want to filter out genes that are expressed in fewer than 10 cells. So again, since we have a list of matrices, we will apply this function to our list. So we create a Sera object for each sample, in which we only keep the exon count matrix, and in which we apply this filter that will be the same as doing this code, this line of code. So we are taking genes that have counts greater than 0 in at least 10 cells. So the created Sera object can contain multiple layers. In this case, we have the counts layer. And we can obtain information in a layer using the @ symbol and some specific data with the dollar symbol. And in addition to the original count matrix, we can store the data that we will generate during all the analyses, such as the normalized matrices or some clustering results, and every time updating our Sera object. So we can have a look to one object. So we take this sample, and here we can see that we have an object of class Sera with this number of features and this number of cells. We have an RNA sequencing assay experiment and only one layer present, which is the raw count matrix. We can also show the number of cells and genes. So the number of columns is the number of cells, and the number of rows is the number of genes. We can also get the names of the cells by applying this function, and here we can see the cell barcodes. And we can get the gene or feature names by applying these functions, so row names or features. And here we can see the gene symbols. And finally, we can also get the count matrix by using this line of code. And so again, we have this sparse matrix representation, or this one. Moreover, the Sera tool offers different functions to assess information. So, for example, to get the count matrix, we can type get assay data by passing our Sera object and the layer that we want to assess, in this case the counts. And for more information, you can go to this link. So next, we need to assign some metadata to our object, because we already have some statistics of our object. So let's visualize the metadata, and here we have the name that we gave to the project, which is the COVID project, and we have the number of counts, so the total number of UMIs for this cell, and the number of detected genes for each cell. But to add some information that could be useful then for further analysis, we could assign the name of the sample of origin and also a label that indicates the sample condition. So if it is a COVID patient or a healthy control. So here we can see that the sample, this sample is a COVID sample. We have in the Sera object other samples with name with this substring heap. So we can leverage this substring to assign the sample group, the sample condition. So our new columns will be called sample and condition. So we assign this data and visualize here again the metadata of this sample. And now we can see that we have the sample accession name, number from Geo, and also the condition of our sample. So we move now to the data quality control, and we can visualize quality control statistics per each cell. So we are going to plot the number of UMI counts and detected genes per cell, and in order to evaluate the distribution of these counts and identify the cells and genes that may cause variation in our dataset. And we already said that the cells with low counts or detected genes might be uninformative, while high counts could indicate the presence of doublets. So we can use this violin plot function to draw a violin plot from a Sera object, in which the features are the number of counts and the number of features. So here we can see the plot, the generated plot for this sample. And we can also visualize the correlation between these two variables. And as we mentioned earlier, high quality cells show higher number of reads and a higher number of detected genes, so a good correlation. And to this purpose, we can use the feature scatter function provided by Sera. Okay, so now we have this correlation that is high, but we can improve a little bit the correlation that we observe between these two variables. And we can do the same for all the samples. So plotting for all samples the total number of UMIs, For each sample, we can plot the total number of detected genes. And again, we plot the correlation between the number of reads, the number of UMI counts, and the number of detected genes in order to identify mainly an upper threshold for subsequent filtering. So we are going to see the scatter plot for each sample. And then we will, based on this plot, we will decide some thresholds to perform filtering of both UMIs and the number of detected genes. So first, we want to remove cells that have an extremely low and high UMI count, since they are considered low quality cells. So let's see, I don't know, the window here with the plot for each sample. And based on this plot, we identified some thresholds that were good for each of the samples. So we found that on average 15,000 can be a good threshold on average for the number of counts. But keep in mind that the filtering should be done for each sample individually. So we can apply this filtering on the number of counts, and we can do the same on the number of detected genes. Even in this case, we found that 200 and 4,000 were good thresholds for almost all our samples, but again, you should decide the threshold for each sample independently from the others. So we with the function subset, we are applying this subset to all our samples. And now we can show the number of cells that we had before filtering and after filtering. So here was the number of cells before filtering, and here the number of cells after filtering. So we can see that we have more or less half of the or less than the initial number of cells. Again, we can visualize quality control statistics for each cell and for each sample. And for example, if we visualize the correlation between the number of UMIs and the number of detected genes for these samples, now we can see that the correlation has slightly improved. And the same will happen for all our samples. And we can observe also then in our metadata that the sample that the counts have been filtered. So if we visualize, for example, the metadata of this sample, we can see that this is the number of counts now is higher than 500 and lower than 15,000, and the same for the number of features, which are higher than 200 and lower than 4,000. So the filtering has been correctly applied. And finally, what we can do is to compute the percentage of mitochondrial and ribosomal genes, and we will filter out cells containing a high percentage of these reads, as they are considered low quality or damaged cells or dying cells, stressed cells. So to compute the percentage of mitochondrial RNA, we can use this function, the percentage feature set. So the idea is that we are going to identify all the gene names starting with the empty pattern. So this will be the name of the genes. And then we are computing the percentage of the read counts of these genes to the total number of counts. And so we call it the percent.mito data. So, and then we will visualize this new data in the metadata of our samples. And we do the same with the percentage of ribosomal reads. And so in this case, these genes represent mRNA that codes for ribosomal proteins. Again, we are searching for this pattern, the RPLS. Okay, so for these genes. And we are computing the percentage of ribosomal genes. Now, if we look to the metadata of our sample, we can see two new added columns that are the percentage of mitochondrial reads and the percentage of ribosomal reads. And now, based on these percentages, we can filter again out some cells. So we can visualize again for each sample. So here for one sample, the percentage of mitochondrial and ribosomal genes, and now we want to filter out for a percentage that is higher than 20%. And so to do this, we again use the subset function, and we take only those cells having a percentage lower than 20%. So again, we apply this subset for mitochondrial genes, and we apply this subset for ribosomal genes. And again, we can show the number of cells for each sample, and also the number of genes that we have then for each sample. So we can see that they are different for each sample. Again, we can visualize quality control statistics, so we can see if we have filtered out these percentages. And here now we can see that the maximum value for percentage of mitochondrial reads is 20%, and also for the percentage of ribosomal genes. And we can visualize the same plots for all the samples. And finally, we can save our Sera object for subsequent preprocessing steps. So with this save RDS function, one thing that I would like to mention is that there are other functions that allow you to identify putative doublets, and that improve the accuracy of doublet detection. And one of these functions is the doublet finder. But it requires a fully processed Sera object. So after normalization, finding of variable features, scaling the data, and running the principal component analysis. And then each cell will be assigned a label, which is a singlet or doublet, and we can filter cells based on this label. So now we are going back to the slides to continue with the other pre-processing steps. So okay, so before starting subsequent analysis, we should ask ourselves whether the differences that we observe in the data are real biological differences or some artifacts introduced by technical noise. So to address this, the first step is to apply normalization to the data that corrects for issues like different sequencing depths or varying library sizes or difference in the PCR amplification efficiency. And normalization ensures that gene expression levels are then comparable across all cells. Next, we need to consider batch effects because even after normalization, some systematic variations can still arise, and especially if samples were processed in different laboratories on different machines at different times and so on. So, to summarize, normalization ensures that differences in gene expression reflect true biological variation rather than technical artifacts, while batch effects represent some unwanted variation that can mask the real biological signals, leading to misleading conclusions and errors in then downstream analysis. Batch effect requires some separated correction strategies that you will see later in a while. So, as we just mentioned, normalization techniques account for technical and cell-to-cell variability and making then gene expression comparable within and between cells. And this step is crucial for accurate downstream tasks such as clustering and differential expression analysis. And a simple and widely used method is the library size normalization that accounts for the sequencing depth. And here the read count of each gene is divided by the total UMI count per cell, and then it's multiplied by a scaling factor. In Sera, the default scaling factor is used in the normalized data function is 10,000. If the scaling factor is set to 1 million, then it is referred as counts per million normalization. And at this point, we have computed the relative counts per cell. This is a second transformation that is needed because in single-cell RNA sequencing data, gene expression values are often highly skewed, with many genes showing low counts and only few having very high counts, as we can observe in the bottom left plot of the raw data. Moreover, data typically spans multiple orders of magnitudes, with genes having low expression and others being very highly expressed. Therefore, applying a log transformation to our raw counts help stabilize the variance across genes and cells, making gene expression more comparable, as we can observe in the bottom right plot. So we have a more compressed distribution, and so the data is more comparable for downstream analysis. We now move to the final step of this first pre-processing part, which is the identification of highly variable features or genes. So after normalizing data, the next step is to select a subset of genes that exhibit high cell-to-cell variation. So the genes showing high expression in some cells and low expression in others. And focusing on these genes help highlighting biological signals inherent in single-cell data and improves then downstream tasks, including the dimensionality reduction and clustering. So defined variable features provided by SERA implements different methods for this purpose. One is a dispersion-based approach that computes the ratio of variance to mean expression for each gene and then ranks and selects genes based on their dispersion values. And there is this variance stabilizing transformation that models instead the mean to variance relationship in single cell data and attempts to standardize the variance across the genes, making them their expression more comparable. And now we will go briefly into the details of this last method. So this variance stabilizing transformation is a statistical approach that normalizes the differences in gene expression and is generally quite robust to outliers and technical noise. And what does it do? So for each gene across all cells, the method first computes the logarithm of the mean and the variance of the expression values. Then it fits a smooth curve, so this smooth line to model the relationship between the observed mean and the observed variance across all the genes. And then for each gene in each cell, it calculates a standardized expression values using the formula that we can see here. So it computes this standardized value from the raw expression of the gene in that cell, its mean expression value, and the expected standard deviation predicted from the curve that we have fitted. So, and finally, it computes the variance of these standardized values for each gene and rank genes based on their standardized variance. By default, in SERA, for example, 2,000 or 3,000 genes per samples are selected, and then they are using downstream tasks such as principal component analysis. A helpful tip suggestion in this case is that selecting too few genes may lose important biological signals, while selecting too many could increase noise in subsequent analysis. Now we will walk through these preprocessing steps in R, again using the SERA package. So. Okay, so we can load again the required libraries. And then we are going to perform normalization by merging the Seurat objects in a single object, but without data integration or batch effect correction, but just following, in this case, the standard SERA workflow. So the idea is to have now a unique object to which we can apply again all the SERA functions. But with this merge function, we will have the count matrices that are kept separated. So we can load our saved object. Okay. And now we can merge all the SERA objects, so all the samples in a single object, and by assigning to cell barcodes the number of the samples, so we know from which sample the cell comes from. So we can now visualize the metadata of this merged object. And so here we can see that we have the identifier, the number of the sample of these cell barcodes, and we will have for the other, the other cells, the identifier of the other samples. So we can show now the number of cells, so the total number of cells of our merged object and the total number of genes of our cell object, of our cell object. So we already mentioned that after filtering, the next step is data normalization. In this case, so the sample-specific differences are properly corrected because we perform normalization for each layer independently, so for each count matrix independently. And so since the row counts are influenced by sequencing depth, library size, normalization in this case ensures that the expression, the gene expression, is comparable across all cells. And then also we will do a more accurate selection of the most variable genes and features. So we can, for example, take the row counts of our first samples, so that are named now counts.1, because we will have a single count matrix again for each sample in our SERA object. So counts 1, counts 2, and so on. So again, we have our sparse matrix, and we can plot the histogram and visualize the histogram distribution of these row counts. So we already said that SERA normalizes the gene expression by dividing the count, the reads of each gene by the total number of UMI counts for each cells, and then multiplies by a scaling factor and applies a log transformation to the result. In particular, it applies the log 1p that computes the natural logarithm of 1 plus x. We need 1 because we have many zero values here. So the default is the log normalize method with a scaling factor of 10,000, and so we are going to normalize our counts. And again, we can show the histogram of our normalized counts. Okay, these are our log normalized counts, and so alternately we can compute the relative counts using 1 million as a scaling factor. And so we will assign it to another object, and in this case the normalization method is called RC for relative counts, and here we are also changing the scaling factor. So okay, again the normalization is applied independently to each layer. And again, we could show so here we have the counts per million normalization. Finally, we are going to identify the variable features, the variables genes, by calculating, using the variance stabilizing transformation, calculating the standardized variance of the gene expression. And because we want to identify the genes that vary most across cells, that are usually the most biologically relevant for downstream analysis, like the principal component analysis or clustering. Here, Sarah first calculate the variable features for each layer, so for each sample, then identifies the common variable features and supplements the list of variable features until the desired number is reached. In this case, the default is 2000. So we apply the find variable features function to obtain to obtain our variable genes, and here we can see the metadata associated to each gene. We can observe that this function computed the mean of each gene, the variance, the expected variance, the standardized variance, and then we have a flag, which is true or false if the gene is variable or not, and the ranking of the most variable genes. And this is for the same for all the samples. At the end, we will have the ranking of the variable features identified for the single, so the individual Sera object. We can finally plot the top 10 variable features, and so taking the first 10 with the variable features function, and use the variable feature plot with the labels of the names of the genes to visualize the plot of the gene average expression and the gene's standardized variance. And so here we can observe that most of the highly variable features, genes, IgH, IgA, are immunoglobulin genes, they produce antibodies that are involved in the immune response. And then later we will investigate in which cells these genes are expressed, for example, in the B cells. And so finally, we can save our Sera object for subsequent analysis. Okay, so let's have 5-10 minutes break, and then we will continue with the second part of the analysis.
Okay, so good afternoon, everyone. I'm Simone Avesani. I'm postdoctoral researcher at University of Verona at the Department of Computer Science. And now we will continue with the analysis and preprocessing of single-cell RNA-seq data. So until now, we have seen 4 main steps of the analysis. So how to construct and import single-cell RNA-seq data in R, how to remove low-quality cells and genes that are not really expressed in our dataset. And also we have seen how to normalize our data and how to extract which are the genes that are more variable inside our cells. So the next step is the scaling. It's called scaling. And this is a very important step for the steps that we will see next in the analysis. And during this scaling procedure, basically what we do is to scale our cells across, scale our genes expression level across cells. So in this way, basically at the end of the scaling, we will have that each gene will have across the cells a mean expression value is equal to 0 and a variance equal to 1. So to scale our cells, our genes, basically we take the original expression value of our gene in a cell, we remove the average expression value of that gene across all the cells, and then we divide it by the standard deviation. So CERAT package, as we have seen before, provides different function to analyze single cell data and also provide the function to scale our data. And one of the most important parameter of this function is called vars to regress. And basically, it allows to remove, if we have some, for example, technical noise inside our data that we don't want to consider in the next step of the analysis, or some noise that could influence the results of our analysis. And basically, using this var to regress parameter, we can specify some source of variation that are different from the biological variance that we want to consider and that we want to remove from our data. So the idea is that with this var to regress approach, we can fit a linear regression model that will consider the variables that we want to regress out as covariance of the model. And basically, at the end, what happened is that we estimate the residuals of our model, and these residuals should represent the new data after that we have removed the variability and the noise that was determined by specific variables. For example, in this very simple example, we include here the M count RNA and mitochondrial percentage as covariate as something that we want to regress out from our data. So how to do it in R? So again, we load the data that we have preprocessed in the previous steps. So we have our CERAT object, in which we have our assay, that is the RNA assay. We have our highly variable features that we have identified in the previous steps. And then we have the different layers, one for each sample regarding the counts, and one for each sample regarding the data. So in the counts, we have the unnormalized count values, while in the data layers, we have the different samples data that are normalized. So what we do to run the scaling is just one function. We need just one function, that is the scale data, and it takes in input our CERAT object. And once that we have executed this function, the result is pretty similar to before, but we can notice that we added a specific layer inside our CERAT object. And this basically this layer is called scale data, and it's a huge matrix that store our scale data. And this scale data, here we have a unique layer for all the samples of our analysis. If we want to regress out some noise, again, we have a parameter that is called vars to regress, and we can specify just one variable or more than one variable if we want to correct for more variables. And it's important, it's mandatory that these variables basically are stored in our Serath object, and in particular in the metadata of our cells. So there exists an alternative approach to normalize and scale the data that is called SC transform, single cell transform. This approach was developed by the authors of Serath package. And this approach, again, is an alternative way to normalize, scale, and identify high variable genes in our data. So the goal of this approach is that, differently from the majority of the state-of-the-art approaches to normalize the data, this approach doesn't rely on the sequencing depth. So basically, the idea is to normalize and scale our data without considering a possible source of variation in our data, that is the sequencing depth. So, and the final goal is that after this normalization and scaling approach, the variance that we have in our data set should reflect the biological heterogeneity of the data. So how it works, our SCT transform. So basically, what happened is that the function that again is implemented in Serath package construct a negative binomial regression model in which we put as covariate the sequencing depth of our cells. And at the end, again, with some other step, we try to identify which are the residuals of these models. And basically, the residuals of these models should be the new expression values. After that, we regress out the impact of the sequencing depth. So to run SCT transform, we have a simple function, again, here. And we can also add some other variables that we want to regress out. And the most, again, we have the parameters that is vars to regress, as we have for scale data. And in this case, I try to regress out also the mitochondrial percentage noise. And what happened is basically that the model, the function try to fit this model and for each gene, and then try to identify a global trend between the estimated parameters between the different genes that we have in our data, and at the end try to define a new function to transform our data in a new value that is no more influenced by the sequencing depth. So this SCT transform function basically will add to our data a new assay. And now I'll show you. So basically, this is our Serath object. And as you can see here, the active assay is no more RNA, but the active assay is this SCT assay. And here we have 3 layers that are the counts, the data, normalized data, and the scale data that are all computed with just one function. And the output of printing the object basically tell us that we have another assay that is called RNA, and that basically store all the previous kind of analysis that we have performed. So when we log normalize the data, I identify high variable genes, and we scale the data. So one very nice feature of Seurat is that of the package is that basically the Seurat object it could be complex, but in some way it allows us to store different kind of results, different kind of analysis that we would like to test and perform on our data in the same object. In this way, basically, it's easy to access different results and to visualize the output. So, as I told you, now the active assay of our data is this SET, but we can easily switch back to RNA, and we can use a very useful function that is this default assay that takes in input our Seurat object, and we specify the name of the assay that we want to set as default. And if now I take the object, you can see that the active assay is, again, RNA, no more SET. And you can see that we have all the 13 layers that we have computed before. While it says also that we have another assay that is this SET available. And again, we can switch to SET just running default assay function. So once that we have normalized and we have scaled our data, we can do principal component analysis. Principal component analysis is a very important step when we manage and we work with data sets that are very big. In this case, we know that a data set, a single cell data set, is composed by many cells. That is, that are the object that we want, with which we want to work. And each of these objects, each of these cells is basically represented by a lot of features, 20,000 and more features that are the genes. And basically, the idea of principal component analysis is to perform a dimensionality reduction of our dataset in order to represent our cells in a low-dimensional space. For example, no more 20,000 genes, but each cell could be represented by 20, 10 principal components that basically explain all the variability that we have in our data. So in general, with this principal component analysis, we compute 50 principal components that are some components that basically represent the variability in our data. And in general, we can say that the first principal component is the one that captures the most of the variance inside our data, and the PC2 is the second one, and so on. So basically, after that, each subsequent component captures the remaining variance of the data. And this kind of analysis, it's really important for us in single-cell data analysis, because with this kind of analysis, we reduce the dimensionality of the data that is useful for the next steps of the analysis, for example, clustering of cells. But it's also very important because we can explore with some plots our data and check if the variability inside our cells, inside our data, basically is determined by some external factor that we don't want to consider in the next steps of the analysis. So to compute this principal component analysis, basically Seurat package provides, again, just one function, so it's very easy. This function basically takes in input our Seurat object. After that, we have performed the pre-processing and everything else. So, and basically, it tries to compute these 50 different principal components in our data. So, as I told you, one interesting point is that if we look at this principal component, we can explore the variability in our data. And now I will show you how to represent our data in a low-dimensional space. So, but before, just to point out that after that we run PCA, principal component analysis, and we print the object, we can see that inside the object something is new. And basically, the Serrat object says that one-dimensional reduction was calculated, and this dimensionality reduction approach is the principal component analysis. So we can plot our cells, and in particular, we can plot, for example, the first two components of our data, of our cells, that basically should represent the majority of the variance in our data. And we can plot them in a two-dimensional space, a simple two-dimensional plot. And this is the result. So basically here, what I'm doing is, again, taking the first two principal components of our that we have computed, and each dot here is a cell of our dataset. And you can see that each cell is basically color based on the sample of origin. So we can see that basically our cells are distributed along the different components. And in this plot, we can say that in general, cells that are close together are similar, looking at these principal components. And cells that are far away are basically cells between there is a huge variance looking at these first two components. And this could be useful, this kind of plot, because, again, we can color our cells using different kind of information. Here I use this group by parameters, and I use the sample information. So basically, again, our cells are colored by sample. But for example, I can color my cells by condition. So we know that we have two conditions, sick and healthy, in our data set. So I can try to check if, we can see some biological signal, if we can see some variability between healthy and sick cells. And this is interesting because you can see that basically along these second principal components, the majority of the cells are red, so are sick cells, while basically in the first component we have these healthy cells. These are basically distributed the majority of these blue cells. And this could mean that probably I will expect some variability between COVID and healthy cells when we will do the next steps of the analysis, or for example, a differential expression analysis. Another interesting thing that we can explore is, for example, if we have a strange distribution of mitochondrial percentage in our cells. And we can use this feature plot to plot our mitochondrial percentage in our cells. Again, we have the first two principal components here. And the idea is that if we see that along some basically components, we have a huge variability due to the mitochondrial percentage, this could mean that we have some kind of noise, or we have some kind of batch effect that could be due to mitochondrial percentage in our sample. And here, the color basically in the plot, I can zoom it. Basically, the color means that it's represented the mitochondrial percentage. So more dark, it means higher mitochondrial percentage. We can do it for ribosomal RNA and so on. We can color for all the features that we have in our data and explore if there is some kind of variability in our data that was captured by principal component analysis, but that maybe is something not interesting for us and that we can consider as batch effect. And maybe we can go back to the scale approach, to the scale step, and regress out that variability. So other kind of plot that we can do is, for example, these width dim loadings. that basically tell us how different, which are the genes that contribute more to the different principal components in our principal component analysis. And a very interesting plot is called elbow plot, and this is very important for the next steps. Basically, here we have on the x-axis we have the principal components, and on the y-axis we have the standard deviation. So the idea is that, as I told you before, the first principal component should represent the higher variability in the data, and is what happened here. And then the variability represented by each principal component basically decrease. And we can see that basically decrease, and for example, between components 15 and components 20, we can see that basically the components represent and explain the same variability in the data. And this is important because, for example, if we want to for the next step of clustering, if we want to extract the real biological signal from our data, maybe it's basically we can avoid to consider all the 50 principal components that we have computed, but it is in this case basically maybe just taking the first 15 that we can see represent the majority of the variance in our data could be enough to have a good result, a good analysis result. So one of the main problems when we deal with single cell data is, as I told you before, is external noise. And this external noise is also called batch effect, and could be due to many different sources. It could be due to different sequencing runs, different technologies, different sequencing platform, or also different pre-processing steps performed for the different data that we are considering in our analysis. So in this, as I told you before, if we want to, extract the real biological signal from our data, we have to, in some way, remove this batch effect, this external noise, and removing this external noise could help us to preserve biological signal and identify. If we are doing differential expression analysis, identify genes that are relevant for us, that could be biomarkers of the disease, and also identify, for example, if we are doing clustering analysis, identify cell population that are meaningful for us in our tissue data. So basically, there are two different approaches to remove this external noise. The first one is the one that I presented before. So basically use this var2 regress in our scale data step, or in our SC transform approach. And there is another approach that is more used if we are dealing with multiple samples, or if we are dealing with multiple data set coming from different technology, for example. And this kind of approach is called integration. So integration, basically, we have allows to combine these different data set, these different samples, in order to preserve the biological signal and to remove the regress out, again, source of variation. So we have different kind of approaches. There are many different algorithms that were developed in the years. Here I reported some of the most famous, for example, Harmony, the anchor-based approach that are CCA, RPCA, and so on. And then we have something that is used also for bulk sequencing, for example, combat, that use some kind of linear model to perform the correction. If you're interested, I suggest you this review that was published in 2020, in which we have a benchmark of the different batch correction method for single cell. So this step is helpful, and again could allow us to remove external source of variation, but it could be also dangerous in some cases. So if we apply a specific integration approach, or if we regress out some technical noise based on some variables that we have that we know and that we have in our data, we can lose biological signal. And in some case, for example, if we are talking about clustering, it could we mean, for example, losing interesting and rare sub-cell population that are in our data, or maybe also cell population that are unique for a specific data set respect to the others. So this kind of integrative analysis and batch effect removal approaches should be used in a way that is checked at the end of the analysis. And here, again, I suggest you another publication that was published last year, and that it's really interesting because they provide some tips basically to evaluate if this integration and integration approach basically is too much for our data, or it's necessary, and if we are losing a biological signal inside our data. So how to perform this integration and batch correction? Again, in scale data, we can use vars to regress parameter. And if we want to perform canonical integration, we can use another function that is provided by, again, Sera. And this function is called integrate layers. The function basically takes in input our Sera object, in which we have different layers, and one layer should represent our samples that we want to integrate, or our different data set that we want to integrate. So basically the source of variability that we want to regress out. And then basically we can specify different integration methods. So Sera basically provides 5 different methods. A couple of them were developed by the authors of Sera, but the others are some methods that are widely used in literature. So, for example, we have these anchor-based approaches that try to let's say that we have 2 datasets, dataset A and dataset B. The idea of these anchor-based approaches is to map our cells of the 2 datasets in a low-dimensional space, for example, using principal component analysis, and then try to identify a couple of cells between datasets that are similar in terms of gene expression, for example, or in terms of correlation in this low-dimensional space. And then use these cells, this couple of cells that are called anchors, to perform a correction of the other cells of our 2 dataset, estimating some kind of correction function that could help us to remove the variability, the source of variability. So usually this integration could take some time, some minutes, and it's something that is computationally intense. So basically, I suggest you to save the data before and after in order to easily visualize and take the results. So, and at the end, basically what we will have is that in our dimensionality reduction result, we will have another dimensionality reduction element that is this integrated CCA, and it depends again, on the kind of integration that we perform and the name of reduction that we want to provide to that. So just to conclude, we can also perform this integration if we have performed before this SCT transform. So basically, we can use integration both with the standard pre-processing analysis, so in which we normalize, scale, and identify variable features, and then we can use the same function to integrate data after that we run this single cell transform. So the only difference is that we have to specify that we are working with this SCT assay, and that normalization method performed was this SCT. So with this is all. We have now completed, the standard pre-processing pipeline to analyze single cell data and to pre-process single cell data, and then in the next webinar we will see how to continue with the analysis, perform clustering, and perform differential expression analysis between our cells. So thank you.